Greetings of the day, students, and welcome back to another study aid video. Today we are going to be discussing the class 9 chapter 5 of CBSE fundamental unit of life. However, today we're going to go each and every cell organelle and the inclusions of a cell in a very short and simplified manner so you guys can understand it very easily. So let's start. Our first organelle is the nucleus. So nucleus in short terms, it's the brain or the control center of the cell, and it takes control over all its metabolic activities, and it's also the biggest organelle in the entire cell. Okay, so this is nothing but now how we say the motherboard is the brain of the computer. Similarly, nucleus is the brain or the control center of the cells, and it controls all the metabolic activities. Activities that involve energy, metabolism, they're controlled by the nucleus, and it's also the biggest organelle in the entire cell. And its secondary support is also doing cell division. Now the nucleus carries all the genetic information of the cell. That's extremely important in cell division, since the nucleus divides first and then preceding the other organelles. So the parts of the cell, nucleus. So here we have four parts. Out of this, first one is the nucleolus. This structure, now this entire is the nucleus. Now this structure in the middle is the nucleolus. Now what is the function of the nucleolus? It stores RNA. Now there are two types of nucleic acids, DNA and RNA. DNA, the genetic part of it, is deoxyribonucleic acid. And then RNA is ribonucleic acid. So the RNA of the cell, it is stored by the nucleolus. Okay, that's the part of the cell. Second, similar to the cytoplasm, there is a kind of mass inside the cell. This mass, or protoplasmic mass, is the nucleoplasm. Then we have chromatin threads. So if you see the substances over here, dark stain, and then they'll be in a thread-like structure. Assume something like this. This will be throughout the nucleus. What is this? It's called a chromatin thread. Now chromatin threads contain the genetic information, dark stained thread mass having genetic information on the DNA. Remember, during cell division, this chromatin organizes itself into chromosomes. The chromosomes are shaped like this. So it shapes itself into these chromosomes during cell division, and then that proceeds to contain the DNA for organizing the cell. Finally, we have the nuclear membrane. Now, similar to the outermost covering, this is basically a protective covering of the nucleus, the outer covering. So this is nothing but the nuclear membrane. So that's it for the nucleus. Let's move on. Then we have the endoplasmic reticulum. This structure over here is the endoplasmic reticulum. So now, what is the use of this and what's the function of the endoplasmic reticulum? So it's nothing but a system of tubules and sheets. Tubules are similar circular small spherical structures, and sheets that serve many roles in the cell. Now this endoplasmic reticulum, it is of two types: rough endoplasmic reticulum, smooth endoplasmic reticulum. Now rough endoplasmic reticulum, right? It usually has these small circle or spherical substances on it that make it appear rough. Okay, in order, if this is an ER and this is a smooth ER, in a rough ER, right, there will be certain substances that are attached to its surface which make it appear very rough, hence the name, or granular. Well, what are these substances? These are nothing but ribosomes. So this part of the ER, the rough endoplasmic reticulum, it has ribosomes attached to its surface, and the ARIA's primary function using these ribosomes is to synthesize and secrete the proteins. This is RER. Then we move on to SER. So this is the part of the ER whose function is to synthesize and secrete lipids. There's no substances such as the ribosomes on the smooth ER. There's nothing on it, just the tubules and sheets. So this part, its function is to synthesize and secrete lipids. Lipids or steroids are a type of lipids, to make it easier for you guys to understand. So these are synthesized and secreted by smooth endoplasmic reticulum. Other than that, what are the secondary functions of the ER? As we saw, RAR synthesizes and secretes proteins, SCR synthesizes and secretes lipids, acts as a cytoplasmic framework. Very important. This transfer of substances is controlled through endoplasmic reticulum, vice versa of nucleus. Or the transport of things out of the nucleus into the cytoplasm and from the cytoplasm into the nucleus are controlled by the endoplasmic reticulum. So this is what it means by cytoplasmic framework. Substances in the cytoplasm are provided a framework and can travel through the ER. Then smooth endoplasmic reticulum. Its function is to detoxify. Now you ingest a poison or a certain drug. The SCR's function is to detoxify that. Remember, detoxification does not mean you can take whatever you want. Harmful substances are highly precautionary. But certain substances, very mild substances to a limit, can be detoxified by the smooth endoplasmic reticulum. Then finally, membrane biogenesis. See, as we'll come on to it later, cell membrane is a substance or a flexible membrane which is made up of proteins and lipids. So, the process of creation of the membrane is done by the ER. The lipids and proteins it secretes and synthesizes are what form the cell membrane, other with secondary purposes. Okay, that's it for the endoplasmic reticulum. Now, Golgi bodies. This is a singular Golgi body. Now, what is its usage? An organelle that prioritizes in package modification and storage of essential proteins and vesicle secretion. Primary function, remember? Package, modification and storage of proteins. So the Golgi bodies, what they do is they take the proteins into vesicles. Again, vesicles are, assume it's a storage type of container. They package them in it and send the vesicles of proteins into the cytoplasm or their destination. That's the primary function of Golgi body. Then, along with this, there are quite some more. Firstly, storage, package and modification of proteins. We just discussed this. Then formation of lysosomes. In case you guys want to know, lysosomes are bag-like structures which carry enzymes, hydrolytic enzymes. These enzymes are used for many purposes. Firstly, to break down foreign cell organelles, that is worn out cell organelles and foreign material. And secondly, if a cell is damaged, it can't spread the infection or the infectious things to the other cells. So that cell needs to be destroyed, and that is why the lysosome does this purpose. So lysosomes are also called suicide bags of the cell. Now these lysosomes are made by the Golgi body. In a simple manner, the ER, right, along with this it secretes a few enzymes, hydrolytic enzymes. They are taken to the Golgi body, packaged, modified and sent out as lysosomes. And cell plate formation. When a cell divides, it needs to form a kind of a cell plate in between, as I call it, in order for it to divide equally into two. So this cell plate formation is monitored and guided by the Golgi bodies. Then we have acrosome formation. Okay, this is something new and interesting. You guys know the sperm, male gamete of the body. In this sperm, you have the tail, middle piece and the head. In the head, between the membrane of the head, there is a nucleus and an acrosome, this substance. For a sperm to function, this acrosome is formed by the Golgi body. The acrosome is formed by the Golgi bodies. And finally, a complex sugar to a simple sugar. Now, a cell cannot directly take in a complex sugar. It needs to break down them in order to ingest it and use it for its energy. That conversion is done inside the Golgi bodies. So that's it for Golgi bodies. Let's move on. Mitochondria. Very popular organelle. The powerhouse of the cell is the mitochondria. Now what is its usage? It's the main site of energy generation. Powerhouse itself means energy and cell secretion. So this is a double membraned. You see this one membrane, two membrane, outer and inner covering, double membrane organelle, and it's very important functioning of a cell. So what is its usage? The energy it generates, right? You can't just call it energy. There needs to be a specific term for it. That term, it generates energy in the form of ATP, also known as adenosine triphosphate. This is the energy that mitochondria secretes. And ATP is also required for active transport or the metabolic transport in a cell. Then main set of cellular respiration. Respiration involves energy obviously, and for the cell to sustain it needs to respire. So mitochondria assists in that. Then powerhouse, through its energy generation, it's called the powerhouse, and primary functioning organelle of a cell. The functioning of mitochondria is almost essential along with the nucleus, the most essential part of it. And without a mitochondria, very shortly cell death is evident without a mitochondria. So that's it for mitochondria, and now we'll move on to the structure of a mitochondria. Observe the figure here. What do you see? We can see the outer membrane, this red covering, outer membrane. Then there's an inner membrane, this yellow substance. That is the IM, inner membrane. Now you'll see that this inner membrane is also called something called cristae. What is cristae? See, in order for mitochondria to produce a certain substance, it needs a lot of surface area. That is, to produce ATP, it needs more surface area in order to maximize the energy generation. For that, the inner membrane is folded like as such. You can see, curve patterns. It's folded as such. That is called the cristae. These inner folds are called cristae. Okay. Then matrix. Matrix is the fluid matrix inside. Basically like a lumen inner covering. And this mitochondria is a semi-autonomous organelle. Do you know why? See, this mitochondria has its own DNA, its own ribosomes. It is capable of inbuilt protein generation, protein manufacturing, and also sustaining by itself due to its own DNA. It has its own genetic information. Now these folds, right, something that's not mentioned here, it contains particles called oxysomes or F0, F1 particles. These oxysomes help in respiration through mitochondria. That's the inner structure of a mitochondria. Alright, let's proceed. Vacuoles. Very simple. It's a storage site of the cell, stores the essential components, and it's a small type of vesicle or a space that's present in animal cells. Remember, a big distinction is that vacuoles are present in both animal and plant cells, but in plant cells, it's a large, very large, occupies up to 90% of cell volume, and in animal cells, it's very small and present in a large number. So the function of this, it stores waste products, stores essential cell exclusions like protein, amino acids, sugars, water, sap. These substances are stored by vacuoles. That's it for vacuoles. And they are single membraned, and they are covered by a membrane that is called as tonoplast. T-O-N-O-P-L-A-S-T. Tonoplast. This tonoplast is what is the name given to the membrane of a vacuole. Okay, so that's it for vacuole. Let's move on. Cytoplasm. See, everything in what we just discussed is a part of a substance. Cytoplasm. It's nothing but a protoplasmic mass inside a cell. And remember, protoplasm means the entirety of the cell other than cell membrane. Cytoplasm means everything in a cell except nucleus. See, this nuclear membrane, everything from the nuclear membrane to the cell membrane is the cytoplasm. And it's made up of the semi-fluid substance. See, everything is somewhat floating, right? The semi-fluid substance is called cytosol. And then other than that, the cell organelles present in the cytoplasm. So the semi-fluid substances is called cytosol. And then all the cell organelles are held by it. And the cytoplasm, the fluidity, right, it allows the transport for intracellular substances. We saw earlier cytoplasmic framework that comes in the cytoplasm. All organelles are present in it. Cytoplasm, simple. Cell membrane, very simple. Outermost covering of a cell. Outermost distinguish it to animals, because in plant cell you have cell wall also. So the outermost covering of a cell in an animal is called a cell membrane or plasma membrane, or a protective covering for both cells. This is a selectively permeable, means semi-permeable or selectively permeable. Only allows transport of certain substances. See, this membrane is very flexible and made up of proteins and lipids. Another reason why cell wall exists for protection. See, in animal cells, they can locomove, they are capable of locomotion and can escape. Cell walls can't, sorry, plant cells can't. So, cell wall is essential for them. So, because of the flexibility in animal cells, it's made up of proteins and lipids. As we saw earlier, biogenesis of membrane, here is where it comes in. And then this flexibility allows for a certain process called endocytosis. Engulfing of food by amoeba using its pseudopodia, that's an example. So a phagocytosis and endocytosis is possible only by flexibility of cell membrane. Now we're done with the cell organelles. Let's take a look at this entire cell structure. We observe a plasma membrane, the nucleus carrying the chromatin, and the chromosomes that are examples of when a cell divides. Here the endoplasmic reticulum, you can see the structure there. Then you also have the Golgi body, vesicles around it. Then mitochondria, the cristae as we just discussed earlier. Other than that, you also see some cell inclusions like ribosomes all over the cell. Then you have centrosomes, lysosomes, the cytoplasm inside. So that's it for an entire animal cell. I hope you guys have enjoyed this lesson and understood it. In case you have any doubts, feel free to comment down and make sure to like and subscribe for more future videos regarding class 9 topics. Thank you so much, students. Have a wonderful day ahead and keep learning.